Welcome everyone to today's Eawag seminar. I would like to introduce to you Siegfried Vlaeminck, Professor Dr. Siegfried Vlaeminck from Antwerpen in Belgium, who will give the talk today. We'll talk about nutrient recovery, not only here on Earth, but also in space or for space missions. Maybe some words about your background. So he basically did his PhD in Ghent, together with Nico Bohn and Willy Verstraete. I think those are names which are known to us, especially when it comes to nitrogen conversion. And that's also the main topic you have been working on. And after your PhD, you continued with an assistant professorship in Ghent, and now you're professor in Antwerpen. And really digging deep into the whole resource recovery and nitrogen conversion topic. And this is also a topic I'm working on myself, so I'm really excited to have you here, and I'm really looking forward to your presentation.
Thanks very much, Kai. Thanks for the kind introduction. Super happy to be here. Thanks for having me. So welcome, everyone. I'll start off with a very brief introduction. So I come from Belgium, and I think most people do know Belgium. We have quite some things in common, like good chocolate, a number of languages, several languages that we speak. But I won't dwell too much on the details. And in Belgium, I work in Antwerp, and Antwerp is known for diamonds, for one of the largest chemical clusters in Europe. So on this skyline here, this is BASF. So you see some Haber Bosch fertilizer production, which is relevant to the story of today. And I am at this beautiful university, which looks old, but the buildings are inherited because the university next year will be 20 years old. So it's a young university, a relatively small university, but a dynamic one, I like to say. So what do we do in our group? It's a bit of a blend between microbial cleantech development, so specific process engineering, and environmental systems analysis, then see how these processes fit into the broader picture and the system. And indeed, as Kai said, we have terrestrial applications, and that's the vast majority of what we do, but also we have a flavor of space in our work. So I like to call it a T profile as our microbial cleantech development. So it's microbial biotechnology is an in-depth focus, and then we zoom out for the environmental systems analysis. And then our group, actually, the microbial systems analysis is co-led by Mark Spiller, also helped me to prepare today's presentation. And then in the microbial cleantech, we have three groups. One is focusing on nitrogen assimilation to go to protein and fertilizers. Another one is focusing on oxidation and reduction, among others, nitrification and anammox. And we have a third new cluster on CO2 and negative emission technologies, which is a bit unrelated to the others. So that's a bit more exotic. And our group is soon to be expanded. And we're looking for one more person, but I'll give you the vacancy in the end. Who knows, you know someone that may want to apply for this environmental systems analysis position to do a PhD in our group. Now, how do we start collaborating with Eawag? It started in 2014, or at least in 2014, we could publish the results of the first collaboration. And Sarina came over to Ghent, where I was at the time, and we did some fish on her samples, fishing for anammox bacteria. We got nice results, and actually this got well picked up by literature. And then with the group of Adriano, around the same time, we co-wrote a review on full-scale partial nitritation anammox, which also was covered quite well. And actually these two are in my top 3 levels of citation. So my best papers are with Eawag. So that's a good reason, I guess, to continue having a good relationship. I don't know, but there's definitely some causality. And then a bit more of an exotic and more recent paper is the fact that we flew Vuna sludge to the ISS and back. So it is the first time urine nitrifying organisms have been in the lower Earth orbit, and we retrieved them, and they were still alive. So that sounds a little bit more exotic, but it was a very fun thing to do, actually. So then the collaboration continued, and today we just finished off the nitrogenizer project together with Kai, and we have 2 ongoing PhD projects on urine nitrification. Now, thus far, just the introduction. Now I'd like to spend a couple of slides discussing some recent review that we did. And there was a review of sustainability assessments of environmental and economic sustainability assessments of nitrogen removal and nitrogen recovery. So the big problem with nitrogen, it's a double-edged sword, so to say. We cannot go without, but we have too many of it in the wrong places. And that's, of course, our environment. And if we look into our planetary environmental problems, you see that the nitrogen and phosphorus are trespassing the safety zone and are in red. So this is beyond, this is a risky area where we are. There's too many reactive nutrients in the environment. And we see that in the black dots, that agriculture is the main contributor to that. And it's a leaky agriculture system. So we should be very careful on how we manage these nutrients, and we should do better. So there's clearly many risks associated to those nutrients in the environment, and those lead up to also genetic diversity and to a smaller part also to climate change. Now, in Europe, we have some numbers on the damage of reactive nitrogen in the environment, and this is between, let's say, 2 and 9 euro per kilogram. So it's a very heavy damage per nitrogen in the environment. Also, if you realize that a kilogram of nitrogen costs you €0.5 if you purchase it. So this is actually, this damage is larger than the benefits that all the farmers get. Now, if we look a bit further into the numbers and into the emissions, we see here what is per person per year going into our fields in terms of Haber Bosch nitrogen and total nitrogen also from animal manure origin. And you see some important pathways through which we're losing reactive nitrogen. Now, if we map this through our economy, and our economy in this case is Flanders, we did a study in 2016, and from the 100 units of nitrogen that we put into our system from Haber Bosch origin, 14 end up in our plate, and therefore in our final wastewater treatment. That means 86 end up in another place, end up typically in the environment. Of course, we can collect a number of those streams, and in Belgium, in our calculations, we have around 11 kilograms of reactive nitrogen that we can treat, that is collectible or already collected. Today, only 17% of that is recycled. So we made a new analysis, which is just published, and that is actually even showing a lower nitrogen usage efficiency of 11%. And in our analysis here, phosphorus also had 14%, and now that rose to 18%. But you see, it's very inefficient as an overall system. Now, just one idea on how we connect in this material flow analysis, all the dots is what you can see here in terms of the origins of nitrogen coming into a sewage treatment plant. And in our case, we have quite some infiltration. The sewers, for instance, our groundwater is contaminated with nitrate, so we also have a nitrate or a nitrogen contribution here. And of course, as you all know, the majority is going to the air. So we have actually a number of nitrogen cycles. We have the big cycle where we start here with actually with our waste nitrogen, if I can call it like that. And of course, in the centralized sewage treatment plants, we make the nitrogen production, nitrogen gas production, the removal, and resynthesize with Haber Bosch to be reused again. Then we have here, I call it indirect reuse. Some people may not agree, but somehow it is a cycle. And of course, then we have the direct reuse, and a lot of it is reused as such, like manure often. And a number of the streams are reused or can be reused after processing, like we have the urine, soil separation treatment, and use as fertilizer, of course. We all know that Haber Bosch is not good. It's linked to a lot of the global energy consumption, a considerable amount of the CO2 emissions. But when we come to look at it, it's quite efficient. It's difficult to compete with it as well. So in the nitrogen use in agriculture, it's providing more than 60% of the nitrogen, and is produced in big scale installations, in fact. So also at the level of the logistics and the scales of things, it's difficult to compete with thousands of tons of nitrogen or ammonia per day. Now, actually, it's of course a very unfortunate situation with volatile prices, but it could give a boost for recovery. As you all know, the natural gas prices have risen tremendously, and so have the synthetic fertilizer prices, which can make recovery more attractive. So, some benchmarking figures, and we have a famous author here in the room. And these benchmarking figures are not perfect, but roughly, of course, if we want to compete with recovery, and this is just one of the features, it's energy and its CO2 footprint, there are many more environmental aspects and things to take into account. But let's have a look here into fossil kilowatt hours. We need a certain number for nitrification, denitrification, and a certain number to produce urea. And if we can translate this in carbon footprint and in costs as well. So, but this is the benchmark, roughly ballpark figures, order of magnitude that we should try to compete with. So let's say together here, let's say around 5, let's say we should be below 10 kilogram of CO2 per kilogram of nitrogen. Now, in this review, we checked what's out there in terms of nitrogen recovery strategies. We tried to group it a little bit. I will not go in detail. The paper actually just got this issue today, so I can share it later also. So it's there for a detailed study. But I'd like to point out a little bit what we did specifically, and that is check how it is documented that recovery is better or not from an environmental and economic point of view than removal. And so we included, we searched with Boolean terms in all the databases, and we included in the end 63 studies. And you can see here, the studies actually had all the different perspectives. Some were looking into recovery versus recovery only, one recovery technique versus another one. Other ones were comparing recovery versus synthesis, but not including the removal part. Others were just recovery versus removal, and only 17 actually looked at the full system view to make the proper full benchmarking. Now, here you see the number of the studies. So 17 ones did it actually as it probably should be done. Now, what came out of this, we saw that quite a number of removal technologies there was some variation, and of course we know that nitrification, denitrification is a typical benchmark, but with the advent of partial nitritation, anammox, we could lower considerably on energy footprint, on carbon footprint, so that has been a reason also, and to make it more difficult to compete with recovery and when your water allows for it. So you can roughly see here the distribution of the technologies in the different papers. Also, and I realize I go a little bit fast, but I like to touch on many things just from high level, and then maybe later we can have in-depth discussions. So what we see here is the typical technologies that were included in these recovery studies and the typical streams they focused on. For instance, there has been a lot of focus on sewage, there has been a lot of focus on urine and on manure slurries. Then you see also as an enabler for nutrient recovery, AD is quite popular, and of course it makes sense as often dewatering or solid liquid separation. But to give you a little bit of the full picture of what typically is being looked at in literature. So let's go to the conclusions. Actually, we had 39 studies that looked at this comparison versus nitrogen removal and synthesis. 24 found that the recovery was sustainable, and then 17 said it's inconclusive, very case dependent, which makes sense, of course. And then there were a number of studies also where recovery did not come out as a better route. So it's not the one against the other. It will be probably a hybrid solution if we want to go for the most sustainable route, but I will definitely include a lot of recovery. So recovery, whether it's sustainable or not, probably, and in many cases, it can be sustainable. It's always difficult in these analyses to compare what you see in a lab or a pilot scale with processes that have been established over decades. And of course, it's a moving target, Haber Bosch, because if blue and green hydrogen are upcoming and are seriously considered for ammonia synthesis, this will lower the carbon footprint considerably. So that is something in our benchmarking to keep an eye on the innovations at the Haber Bosch level. Good. We're approaching the technologies. And in terms of recovery, I'll be touching more specifically about recovery as nitrate and recovery in microbial biomass. And actually both can go in two directions. They can be used as fertilizer for plant production, and they can go into the direction that I like also very much, and that's about producing microbial biomass for the purpose of feeding typically animals, could also be plants. We'll get to that. So, some solutions now that are based on nitrification and a partial nitritation anammox. Now, many of you probably know mainstream deammonification, or what we call cold partial nitritation anammox. If we have the mainstream sewage treatment line, and there's quite a bit of work that happened on this here at Eawag. If we pre-concentrate the carbon, and then we can send it to the digester, and then have a carbon-lean way of dealing with the nitrogen, or with the residual of the nitrogen, we can tip the energy balance, we can go to energy autonomy, etc. So what we want is reduced energy consumption and increased biogas production, increased energy recovery, and then tip the balance to become at least autonomous, grid independent, perhaps even slightly positive. And that should be more sustainable and potentially cheaper. It depends a little bit. So there's a conceptual framework on how to achieve this nitrogen conversion, and you can see it here. It's coming from a review that we published a while ago. So in green, you have the bacteria that you like and that we want to see, and that's of course anammox bacteria, the ammonia oxidizing bacteria, and the heterotrophs that are denitrifying starting from nitrate. And in red, we need to kick out the nitrite oxidizing bacteria and the aerobic heterotrophs. We should minimize the aerobic conversion of COD. And we're not against denitrifiers that use nitrite. So any shortcut route for nitrogen is good if we can channel away the COD in an anoxic way. That's advantages for our energy demand. So there's a number of strategies, but it's not easy, the recipe for mainstream anammox. And we have a number of strategies needed that all need to be combined and that are based either on retaining the desired ones or co-seeding them and washing out of expelling the undesired ones, and further on promoting the kinetics of the desired ones and suppressing the kinetics of the undesired one. Now, we concluded last year a long-running pilot experiment at the wastewater treatment plant in Breda, which is one of the 8B plants in the Netherlands. There's 3, if I'm not mistaken. And this was done in collaboration with quite a number of people and instances for the funding. And to summarize a little bit, we had a multi-stage type of pilot where we had a pre-denitrification, and then we had an anammox tank which received the side stream partially nitrified water, then go to intermittent aeration, and then we have a continuous aeration tank that is just making sure our settling is functioning properly. So here, intermittent aeration, we have ideally the full partial nitritation anammox together with nitritation, denitritation ongoing, while here we give a boost to the anammox bacteria, and here we make sure that COD is preferably used to remove nitrate rather than being oxidized further on. We also have a physical stimulation to keep anammox inside with a selective retention of the larger granules to make sure they stay in the system, and we have a split SRT, high SRT for anammox and lower SRT for the NOB. Now, This is an example of some of the performance. So it ran over more than 2 years. And you see here effluent levels that were sometimes good and sometimes less good. So TN levels are too high to be discharged. But if we focus on some stable periods where there were no technical issues, for instance, this summer period and this winter period, we had reasonable rates comparable with the conventional B stage, and there's still some fine-tuning to do with regards to the effluents. And one reason for that is that we worked always at a considerable ammonium residual. You can see it here in green. So that is a way of keeping the AOB stimulated and to keep them just a little bit ahead of the NOB.
Just give us an idea about the influent level of TN.
That should be 50 to 60, if I'm not mistaken. Yeah. So, and our discharge limit is 10 in that case, but also that is a moving target, as in some plants and big plants, it's upcoming that we'll need to go lower, which is still challenging and which may require for some of these final applications definitely a tertiary treatment as well. So our technology was COD-lean. The current B stage is consuming 6 grams of COD to convert a gram of nitrogen, while our systems were in the range between 2 and 4, let's say. So more COD-lean and more energy efficient, but not perfect yet for the next step. We did some calculations in terms of the energy balance of the whole plant if we now would upscale this and what would be the repercussions. And of course we could direct more carbon to biogas, and this comes with an additional gain of energy. We would save on electricity on the B stage, and at this moment it's an AB installation not meeting nitrogen limits in the B stage because there's not enough COD. So there's a huge effluent recirculation back to the A stage, and the A stage is partly denitrification because we need to get rid of that nitrate. And so there's a lot of pumping going on to get that going. So you see, if you can save on that pumping, it's also a big cost. Then of course, some of the mixing and sieving has an energy cost as well, but still, it's of course a desktop extrapolation. But these savings could correspond more or less to a bit more than the full electricity consumption. So it can, if it would work and if the water will be dischargeable, really contribute to that.
How large is Breda?
More than 300,000 people. Yeah. It's a reasonably large system. Then we did a study, much smaller scale in the lab, and we saw that we have quite some issues with anammox in the mainstream in the cold temperatures. So we investigated whether we can harvest in summer, store without additives, no nitrate, no redox stabilizer, no energy costs to cool, store for 6 months. Of course, we lose some activity, and then reactivate to bio-augment in principle in winter. And we showed in the lab that this was a feasible route conceptually, at least. I'm not showing numbers here, but just we can discuss if we have time, but we recently published these findings as well. Okay, they are moving around. And then another concept is a little bit linked to what we did in the pilot, but we wanted to study this more. So if you want to do proper bio-augmentation is not always easy. If you from outside bring bacteria into a new niche, you may lose them in any case. So inspired by the BAIT concept for nitrification, we wanted to create somehow in-between conditions, in between side stream and mainstream, to grow and boost the right anammox bacteria at a temperature and nitrogen conditions that are closer to the mainstream than the full side stream. So we call it our nursery concept, where we settle off the granules, or we sieve them out, and we boost them with the ammonium nitrite coming from the side stream. Like this, give them a good boost, let them grow a little bit before we can again put them to the more hostile conditions in the mainstream. And this also seemed to be quite well. It's a good concept, as we showed in the lab. We saw that there was effectively not only an effect of the higher temperature and Arrhenius, but that also, and not only of the higher nitrogen levels, but that also the matrix as such, with a bit more conductivity and salinity, probably gave a boost to anammox under these conditions. So it's a concept that we're working on further. Now, moving away from the mainstream and from the Earth, we go briefly to space. And space today is a very linear economy. More and more initiatives are looking into circularity, even recovering the shuttles, like has been done by SpaceX. There's only one example at the ISS of circularity, or the key example is the water circularity that is currently ongoing. Now, in Melissa and the future case of space long-term human flights, we need much more regenerative systems. Otherwise, it will be too heavy to launch. It will be too bulky to transport such a long distance. So this seems very exotic, but it can inspire us also for terrestrial applications. And in space, if we look where is the nitrogen, it's virtually all in the urine. So if you see what is coming out of a crew member, as you know, it's limited in terms of fecal matter, the nitrogen content. It's mainly in the urine and a little bit in other sheddings of, let's say, our body, whether it's then skin, hair, nails, etc. And then we have a bit more of nitrogen looping into here, a food production system, and part of it part of this protein is in inedible parts, part is in edible parts. Now, within this framework of what we call Melissa, European Space Agency is looking into a number of routes on how to deal with nitrogen. Firstly, you need to stabilize it to mitigate the risks. So that's one aspect that always needs to be fulfilled. If you don't do anything with the nitrogen, we'll have a conversion into spontaneous ureolysis and volatilization of ammonia, which in a very confined compartment is dangerous, of course. And if we don't stabilize it also, it will get some biofouling, will get growth on the organics. So any system should at least comply with stabilizing to mitigate the risks. And then we have two, let's say, classes of solutions. One class is based on full or partial nitrification. Partial nitrification, that's of course the work also of Valentin and Nele and of Kai. And then the full conversion to nitrogen gas is what we jointly did in the nitrogenizer project. And both technology classes allow for water recovery, which is very important. And we tested also if it's an option to go to membrane-based process, because today it's a heat-based process in the ISS, which is very energy intensive. And of course, if you work with RO, you want minimal ammonium content in the water, so both can live up to that. And then if we go over gas production, the typical route that today we see for this gas is that the spaceships and the habitats are leaky. There's always gas escaping. So this artificial atmosphere, either you bring extra bottles to supply and resupply, or you produce on site. And of course, for quite some crops that are high in protein, the legumes, the pulses, leguminose plants, biological nitrogen fixation is a route. And as we know from the work of Kai and Vuna, then the other route is of course nitrate-based fertilizer solutions. So very briefly, what did we do in nitrogenizer? We used a zero gravity compatible aeration technique, which is the membrane aerated biofilm reactor. We bring in the oxygen, get a biofilm here with partial nitritation anammox to form the N2 and the CO2 in the urine. And then we added a membrane to extract the gases, as that's also important in the concept of the space applications. we can achieve, this is a bit how the setup looks like in the lab today. It's not optimized for scale-up yet, but with this extraction membrane, we can effectively extract most of the nitrogen that we are producing in the system. And today we have pretty good conversion rates of more than half of a gram of nitrogen per liter of day of our reactive system. So the rates are good, and we work on real urine at the 10%. It's a 10% urine, so it's diluted with water. So inspired a little bit by what Kai has been doing and what's occurring in Vuna, we also looked into other sources of nitrogen to nitrify to get a liquid nitrate-based solution for plant production. And this is one example here in a recent study where we looked at organic fertilizers, which are currently not, let's say, accessible if you want to grow in hydroponics. You need liquid-based fertilizers. How easily we can convert them into a liquid nitrate-based fertilizer with a simple SBR type of system. At 35 degrees, we reached today around 200 milligrams of nitrogen per liter per day as conversion rate, which is still a little bit on the low side. And then we also did the calculation on how we should supplement to get a complete fertilizer that is mimicking what we're typically expecting in a hydroponic system. And then we did some cost estimates, and actually, if you compare it to buying or purchasing some of these liquid-based nitrate fertilizers for hydroponics, we may have a bit of a business case, at least at the consumable levels, at the operational levels. We have not quantified the capex, but we need to work further on improving the rates. I'd like to share this concept with you. And inspired by it, we're also today looking into making a nitrate-based solution based on nitrogen that is either stripped out of a slurry or that is coming in from ammoniacal air like you have in animal stables. So the idea is to replace the chemical acid scrubbing that is relying on the chemical acids, and instead our goal is to be cheaper and to be less dangerous also for the operators. You don't need to purchase the acid, it doesn't come with the footprint of these that they typically come with, and to in situ produce acid and nitrate. So to make use of the protons produced to scavenge the ammonia in the scrubber. So this development is ongoing. So maybe next time I can show a bit more on how this is working. And I'd like to finish off with something exotic that we've been doing in this area. And we wanted to see if we could push up the boundaries of nitrification and all the other nitrogen conversions. And this, the textbook always said, yeah, if you go above 35, everything stops. And then we were curious what's happening above 40. And we actually, in the framework of the PhDs of Emily and Tom, could set up nitrifying reactors at this high temperature, which required AOA. So we find ammonia oxidizing archaea there. Denitrification is not difficult. It was quite a bit of a challenge to combine both in one system, nitrification and denitrification, to get it properly tuned. Then more recently, we also could establish anammox around, now I forgot the temperature, but I think it was around 50 degrees, the exact temperature. And we hope it's upcoming maybe later this year that we find a new organism, a new type of anammox that is functioning under these high temperatures. So currently today at Ghent University, I am co-supervising, but the lead is with Jose and with Ramon. There's two people still investigating on now whether we can merge partial nitritation with anammox in a system. So let's see. Our goals is, of course, first curiosity, can we run these systems in a reactor setting? Secondly, there are quite some warm effluents. If we have thermophilic digestion, there is an advantage in terms of rates and a number of things. So if the heat is present, and then if we have a faster process, we could get to some cost savings as well. But that will take us still some further years to see if that's indeed the case. And now I'd like to go to some other recovery solutions that are based on protein or microbial biomass to be used as fertilizers. So what is microbial protein? It's actually used for single cell protein, and it means protein embedded in a cell. So biomass rich in protein, as most microbial cells are. And how do we produce them? Generically, of course, you start with an inoculum, sources of carbon, nitrogen, phosphorus, electron donor, acceptor, etc. You have a bioreactor production, you harvest it off, and what is typically then done is drying. That's it. It's dried microbial biomass, dried sludge, if you want to say so. If you use it in food and feed applications, we call it microbial food feed, or typically also microbial protein. And if we go into the root of fertilizer, organic fertilizer applications, which remind of the biosolids approach, which is not linked to high rate production, but it's of course extremely stabilized sludge already. So there's a number of categories of organisms that are currently being investigated, and so you see 5 groups here, and I'll talk today about what I call ordinary aerobic heterotrophs. They're ordinary because they're not C1 oxidizers, so they are not methylotrophs. I won't talk about the hydrogen oxidizing bacteria, but briefly touch on the microalgae in also the context of space and urine nitrification, and on the purple non-sulfur bacteria. So this field of microbial protein has been, let's say, it's a bit of a renaissance. There has been a wave in the past of this, then due to various reasons, it didn't break through, but it seems now today there's a new wave. Cost aspects are different. The needs for sustainable production are quite different. And you can produce microbial protein in several routes. You can use primary resources that ideally are clean. Today there's also companies using fossil fuels, using natural gas to make microbial protein, which still environmentally can make sense compared to land-based production of protein. But our interest is in clean production, and as is linked to wastewater here, we're actually in the right column, based on secondary resources, typically on wastewater-based resources. And that, of course, that excludes us from using also our biomass as a food ingredient. So we're looking into feed ingredients. There's a number of requirements if you look into this area, and I won't go into the details, but like with any process, you want to check out and make sure you go for minimal impact on the environment. You go for a system that legally can make sense, one day at least, and then there's many other things to take into account. But let's first touch on acceptance. So we went out on the street, very small sampling. We say, are you willing to eat microbes cultivated on wastewater? Actually, the people in Belgium, it was not too problematic. A third said, okay. And then we said, okay, but the quality will be definitely safe. And then up to half were definitely sure, and another 20% said, yeah, okay, maybe. This is not properly executed science, and this is what was done pre-corona. So I don't know what the term microbial now is creating in terms of perception with the people, but it's something to be studied definitely. Now, if we want to produce microbial protein for animal feed, we typically look at food and beverage effluents. So for instance, we look at breweries, potato processing companies, yeast companies, any type of other starch, dairy, etc. So these are, you could call them feed-grade effluents. They're not contaminated with human pathogens, at least. And a typical example of the 3 types of microbes that we're looking at, the aerobic heterotroph, you know them, it's a typical activated sludge type of system, but it's a high rate activated sludge type of system. Maximum conversion of COD to biomass, to good proteinaceous biomass, and we need a secondary treatment to finish off to dischargeable effluents. And then we have another route after the digestion, after removal of the majority of the carbon, go to the microalgae, or use another phototrophic route with the purple non-sulfur bacteria that really thrive on volatile fatty acids. So if we pre-ferment anaerobically our water and then with the VFA go into the PNSB production, then we also have a route, and then the rest of the water or the remainders can go into anaerobic digestion. So in a sustainability assessment, in an LCA that we performed on this, we benchmarked these products based on potato wastewater with soybean meal, because we're importing, at least in Flanders, but the whole of Europe, a lot of soybean, particularly from South America, so sometimes from other regions. And at the level of the human health, let's say we're at half of the impact of this soybean meal, and it's even better for the ecosystem's impact, so less species lost in this case, but we were pretty high for all three on energy, and the lowest on energy was microalgae. That's linked to the fact that you can have full AD before the anaerobic digestion still allows full energy recovery before you have the microalgae production. So there's still some work to be done at the level of the energy and the resources used there. And one route is, for instance, avoid drying, or and work with a paste, stabilize that paste and work it into your final product, or use alternative sources of heat. Just for the sake of the exercise, we also looked into sewage and wanted to see whether it could make sense in terms of flows of carbon to go to a number of these products. And we looked in the EU28, sorry, excluding Switzerland, into maximizing COD capture, bringing it to the sludge line, maximizing the release of carbon with a thermal hydrolysis, and then VFA production. And they go in three routes. One is esters, the other one is bioplastics, and the other one is protein. And in red, you see the global market, and in green, you see how much we could produce based on the sewage flowing in Europe. And actually for protein, now I need to, so what we see here for protein is in red, this is the global market of fish meal, and in blue you see what we could produce in Europe. It's a thought exercise. You see also for bioplastics, we can easily recover more than is present on the global market, and definitely the case for esters. Now, if we go to plant production with this microbial biomass, then we call it microbial fertilizers. So they're a type of slow release organic fertilizers, but slow release is relative. Over a period of, let's say, 3 months, they release up to 70% of the nitrogen. So it's a good amount of nitrogen that is available over the growth season. So similar pre-process, we produce biomass, then we dry it, and then it can be blended so that the macronutrients are present in the right proportions. And then the idea is that the soil is responsible for mineralizing this biomass, for releasing the nutrients, for nitrifying, and then so that it is available to the plants. Now, we did a preliminary assessment of the sustainability of this and compared it also to other organic fertilizers that are plant or animal based and to mineral fertilizers. So there's a lot on this slide, but what we see here in the beginning are the microbial fertilizers, then a lot of conventional organic fertilizers, and then the end, a mineral fertilizer. So what we did here is make a blend of the N, P, and K inside. So the functional unit is a blend, and it's all corrected for this, so that we can normalize somehow, because all the individual fertilizers have a different profile. So it's normalized for that. And we saw that it's actually, with a conventional LCA, for most of the organic fertilizers, not easy to compete at the level of human health, ecosystem quality for some of the organic fertilizers, others then again that was easier, and the same for resources. So with basic LCA methodology, we could not show organic fertilizers that they are more sustainable than mineral ones. And if we here included carbon and we say we'll sequester in the long term 10% of the organic carbon, those to the soil, you can see everywhere it's lowered a little bit the impact, but it's not very easy to reach the metal fertilizer ones. Good. I'm looking a little bit at the time, so feel free. How much do we have more or less round up?
More or less.
More or less round up. So I will maybe just go to the point of some nice features linked to our microbial protein studies that are not at the level of the production, but resource recovery is also at the level of the use of the biomass. And we grew microbial protein based on real brewery wastewater, and we use it in a nice feeding trial with rainbow trout. And we replaced up to 75% of the fish meal, and still we had similar feed conversion ratios, so you don't need more feed per amount of fish produced. And the fish grew at the same rate, and they had similar weight gain. So if we can replace up to 75% of the fish meal, this is a big impact in terms of sustainability. And maybe I'll finish off with a very similar story. If we look at purple bacteria, if we grow them on wastewater, in this case from a monastery, from a Trappist brewery, you see there's some consistency in our work. And we then go and feed shrimps. We had actually even a decrease in FCR, so we needed less feed per gram of shrimp produced, and we had a bit of an increase here on the amount of biomass produced over a certain amount of time. So these microbial fertilizers, microbial protein can really, is really not a disadvantage if you are producing animals. So I'd like to thank the full team. This is not all my people, this is just a full department. All our funders, I'd like to thank you for your attention, and I'll maybe just keep this slide on. If you're interested or you know someone looking for a PhD, they can always get the data here or get in touch with me. Thank you.